When dogs poop, they don't poop pure DNA. So when a sample comes into Mr. Dog Poop's lab, the first thing we have to do is separate the DNA from the actual poop. The process begins when the dog poop tech puts the sample into the dog poop centrifuge. The sample will be spun at about 4,000 times normal gravity for around 4 minutes. This will separate out the solids and the solution that we use to preserve the DNA during the transport process. Once that process is completed, the dog poop tech will take samples from different sections of the dog poop and put it into tubes that contain little glass beads and lysis buffer. 200 milligrams is used in each tube. Several tubes will be needed in order to collect enough DNA to facilitate all the tests that have to be done. This is a lengthy process, and it's important to remember when collecting the dog poop that you need to send as much as possible. The dog poop may contain hairs or foreign matter, grass, sticks, stones that don't contain DNA. So always do your best to collect as much DNA as possible. Now that that step is completed, they'll go into the cell disruptor. The cell disruptor will shake the glass beads and the lysis buffer and poop in the tube around 3,000 RPMs for a period of 5 minutes. This will separate the DNA from the cells. Once that's completed, they'll be removed from the cell disruptor and put into a centrifuge and spun at around 10,000 times normal gravity. Again, this will separate out the solids, the glass beads, and the poop, and leave the DNA on the top. After a minute in the centrifuge, you'll be able to see the separation of the solids. Bottom is glass beads, the poop, and the DNA is on the top. Now the DNA lab tech can extract the DNA from the solution and put it into a filter. That filter will be used to separate out all the poop particles that are still floating along with the DNA. This is the first in many filtering processes. If you look at the filter is full of poop particles and the DNA is in the bottom of the tube. Now the filters are discarded and we'll go into a second filtering process. To do that, the lab tech will have to add a binding buffer. The binding buffer will make the DNA stick together like spaghetti in a pan. By clumping the DNA, it won't be able to pass through the filter, and we'll be able to get all the other particles out of it. The binding buffer is added in multiple steps and then mixed thoroughly with the DNA. Once all of those are completed, half of the solution will be taken and put into a spin filter, which will go back into the centrifuge. Now we've filtered out all the impurities and we have the DNA on the top of the filter. This allows us to go into the washing steps. We use wash buffer to wash the clumps of DNA. This will happen several times, again going back to the centrifuge until we do an elution step. The elution step will give us a little drop of actual solution with DNA. We'll go through one more filtering process, a final filter, and now we have pure DNA. That DNA will be moved to a cryo tray and then moved to a freezer where it can be stored at temperatures as low as -80 until it is ready for the next step in the process.